Welcome back to your Chem 4 lecture. We are moving on in chapter 17. We're going to talk a little bit more about translation now. So we talked about what transcription and translation mean in the previous lecture. Now we're going to dive into how that actually works at the ribosome. So the tRNA basically is going to convert the message from the mRNA into a protein language with individual amino acids. Okay, so this is how it works. The genetic code, as we know it, basically is a series of 3 nucleotides, or what we call triplets, in the mRNA. This is known as a codon. So a codon is 3 nucleotide triplets from mRNA, and that specifies the amino acid, and it also is going to tell us what sequence it's going to have in the protein. So for example, if you had 9 U's in a row, the way this would be read is the first 3 are UUU, that's a triplet or a codon, and that codes for phenylalanine. UUU again codes for phenylalanine. UUU again codes for phenylalanine. So you get 3 phenylalanine amino acids in a row in the amino acid sequence in a polypeptide. So there's codons for all 20 amino acids, and there's also what's called a start and a termination codon as well. So at the beginning of any mRNA, the codon AUG is going to signal the beginning of protein synthesis. So AUG is your start codon. And then there's 64 codons possible total from A, G, C, and U. Imagine basically it's just 4 times 4 times 4. So that gives you 64, as you know. And then we have stop signals at UGA, UAA, UAG to tell us that the protein synthesis is ended. So this gives you, your start, and then however many amino acids are in the polypeptide, and then end. Because both of those are important, right? If you didn't finish when you need to finish, you didn't start when you need to start, you're not going to have the right protein. So here's the table. Now, of course, you don't have to memorize this table. This is going to be given to you on the test, but you do need to be able to read the table. So if you're given a triple, like let's say I give you the triplet, or I should say codon, A, G, G. What you do first is go to the first letter A. Okay, so we're here, second letter G. So AG and then AGG is here. That codes for arginine, or R. So you know then that the first, or the amino acid that's relevant there is R, or arginine. Likewise, let's say that I gave you the codon CGU. Okay, CGU, what do? We go C first as your first letter, then G as the second letter, and then U. Oh man, I can't come up with anything new. That's still arginine. So how about that? So that's how you can read these codons to know what amino acids are being formed. And then again, you have your start codon available right here, AUG, which is also known as the methionine codon. Okay, so let's say that we gave you, let's say that I gave you CCU, AGC, GGA, CUU. And I asked you what amino acids structure are going to follow from the codons that are given here. Well, first you split it up into 3s. So CCU is first. If we look up CCU on the table, CCU, that codes for proline. So proline is going to be number 1. AGC, we go back here, AGC, serine, GGA, glycine, and then CUU, leucine, just in the same way. So we know that the codon sequence here, CCU, AGC, GGA, CUU, is actually proline, serine, glycine, leucine. Okay, now why don't you go ahead and give it a shot. Now I'm not going to make you hunt through that table. I actually give you a little small sequence here, but if I give you codons in this manner, GCC, GUA, GAC, what amino acids are going to be coded by this section of mRNA? So go ahead, pause the video. When you come back, we'll take a look at the answer. So pause. Okay, so GCC codes for alanine here. GUA is valine, and then GAC is aspartic acid. So ALA, VAL, ASP as our amino acid sequence there. Okay, so we're going to talk briefly about transfer RNA now. So tRNA, we mentioned in the previous lecture, contains an anticodon. This anticodon is exactly what it sounds like. It matches up exactly with the codon from mRNA. It's a triplet of bases. Basically, those three come down. You have your three mRNA here, and they match up in base pair just for a moment during the translation of the RNA. There's also the acceptor stem. This is what's going to form an ester bond with an amino acid. So activation of tRNA just means that you take the amino acyl tRNA synthetase. That's the protein, or the enzyme, I should say, that catalyzes this reaction. There's an ester bond that forms between the carboxylate group of the amino acid and the hydroxyl group of the acceptor stem. Okay, so let's go ahead and start by going through the processes of protein synthesis. So these are the steps. So initially, the beginning step of protein synthesis is what we call initiation. Protein synthesis starts when the mRNA binds to a ribosome, and then the first codon in mRNA is the start codon, and then that forms hydrogen bonds to a methionine tRNA. So that's the start codon, codes for methionine as an amino acid. Then a tRNA is going to bond to the next three amino acids, so the next codon, and that places a second amino acid next to the methionine. Then the ribosome catalyzes the formation of a peptide bond between the C terminal of methionine and the N terminal of the next amino acid. So the amino acid is being built from the C terminus to from the N terminus to the C terminus. So yeah, this is what an activated tRNA would look like. Again, just has the same cloverleaf structure that we looked at before. The codon on mRNA is matched by an anticodon on the tRNA at the very tip of this clover here. If your codon is UCA, then the anticodon would be AGU, and that would code for serine. So this is why UCA codes for serine. It's because the relevant tRNA anticodon is the serine tRNA. Okay, now the next step is translocation and chain elongation. So the first tRNA now basically moves on in the ribosome and allows it to detach, and that basically shifts the whole RNA complex over to the next codon. This is what's called translocation, the movement of the mRNA in the ribosome. Now we're in the process of chain elongation, because this process is just going to repeat again and again. New tRNAs can then attach to the new codons, and new amino acids are going to be linked to our our elongating amino acid chain, and this process continues apace as the amino acid chain gets longer. So this is what it all looks like here. So this is in your book. You're going to definitely have a better time looking at this in your book. But now we're kind of looking down here. This is how we draw the ribosome. We just imagine we have two spots here, basically. There's this spot here, and then the second spot. So this was where we started, and then we had the new codon come in here. So this is our new tRNA. It's going to add now isoleucine to this chain here. So alanine and isoleucine are going to form a bond, and the growing protein chain kind of forms a long chain behind it here. Right, and then once this moves along the chain here, so the next one is going to be a GUC codon. So once we make it there, then this tRNA has given up its protein chain and it's ready to leave. Oops, there we go. So finally, eventually, as this continues, the amino acids are going to hit a stop codon. Sorry, the ribosome hits a stop codon. That's UGA, UAA, or UAG. There is no tRNA that has an anticodon for the stop codons. So basically, the ribosome gives up at that time. That's when the polypeptide gets detached from the ribosome. And then also, usually they recover the methionine at this point through another series of enzymes so that it can be reused for the next protein chain. Because methionine is always used at the beginning, but then it's not really used in the middle anywhere. Because again, it's just there to start things off. Oh, and this step is known as termination, I should mention. So once the polypeptide gets released, we have R groups on the amino acids in new polypeptides that can form hydrogen bonds to give secondary structures for alpha helices, beta pleated sheets, or triple helices. So there's some of this that occurs on its own. Also, there are kind of helper enzymes in the area that allow the formation of salt bridges and disulfide bonds, building up into an actual protein structure. So a lot of times there's helper proteins there to help fold these proteins. So this is our overall steps of protein synthesis overview. We start from DNA transcription. That's where DNA produces mRNA. Then we have tRNA activation, where tRNA picks up specific amino acids for the given anticodon. Then we have the translation of the RNA, which occurs in the ribosome. And that's where mRNA binds to the ribosome. And that's where we have translation. Initiation first with the methionine tRNA with the start codon. And then chain elongation, that's where new tRNA come in for each additional codon. And that's where we grow the protein chain at that time. And then finally, chain termination, that's when we hit the stop codon in an mRNA. There's no more transfer RNA that matches up to it. So you're no longer building any protein chain. The protein gets released from the ribosome, and then it's folded and it's over. Okay, also coming up with our complementary sequences, remember that DNA has a base pairing with its template strand and its informational strand, either way. That can then be translated into mRNA, or I'm sorry, transcribed into mRNA. mRNA gives a series of 3 codons here, so GCG, AGU, GGA, UAC. So the way we're looking at it here is the template strand then pairs up with the mRNA is what we're thinking about. And then that mRNA has anticodons on the relevant tRNA that code for particular polypeptides. So GCG as a codon pairs with CGC as an anticodon, which carries with it alanine. AGU as a codon pairs with UCA as an anticodon, carrying serine. GGA turns into CCU, which is glycine, and then UAC and AUG pair to form tyrosine. Okay, so let's review what we learned in this lecture with the sample exercise packet. What I want you to do first is pause the video here and match each of the following terms to a definition or concept. One is activation, then initiation, translocation, and termination. So talking about the different steps of translation. Go ahead, pause the video here, come up with your answers, and we'll take a look at them together when you come back. So pause. Okay, let's take a look. So A, ribosomes move along mRNA, adding amino acids to a growing peptide chain. That is clearly translocation. Again, this is when ribosomes move, is translocation. A completed peptide chain is released at termination. A tRNA attaches to its specific amino acid. This is activation of the tRNA. And then a tRNA binds to the AUG codon of mRNA. That's the start codon, so that is initiation step. Okay, sample exercise number 11. The following section of DNA is used to build mRNA for a protein, GAA, CCC, TTT. Give me the corresponding mRNA sequence, then the anticodons for the tRNAs, and then the amino acid order for the peptide. This is the big question that you're going to see on the final. So make sure that you're paying attention here, come up with your answer, and we'll take a look at the answers together when you come back. So pause video. Okay, I'll assume you worked it out. Let's go ahead and take a look. The corresponding mRNA sequence, G pairs with C, A pairs with U, A pairs with U, then CCC becomes GGG, TTT becomes AAA. The anticodons here would be GAA from CUU, CCC from GGG, and UUU for AAA. Then we look at the codons to give ourselves the amino acids. Again, anticodons, we don't use those as our structure, but we know that CUU as an anticodon carries with it GAA as the anticodon, carries with it the amino acid leucine. So CUU as a codon translates to leucine, GGG translates to glycine, and AAA translates to lysine. And again, you have to go back to that chart that I gave you at the beginning of the lecture to get that. Okay, so we'll talk about this real briefly, just about antibiotics. Antibiotics are really useful because they basically stop the synthesis of proteins in bacteria. So we'll talk about a few different proteins here. Chloramphenicol, for example. And what this does is basically it stops peptide bond formation for tRNA. Erythromycin inhibits peptide chain growth by preventing translocation. So it basically messes with the ribosomes. Puromycin, and ribosomes of bacteria are different from our ribosomes. So that's why you could inhibit bacterial ribosomes and not worry about your own ribosomes dying. Streptomycin prevents attachment of tRNA, and tetracycline prevents binding of tRNA. So in all of these ways, it's able to mess with the protein synthesis in bacteria. Okay, so final sample exercise here for this lecture. Give me the following statements in the order of their occurrence during protein synthesis. So this is basically a mixed up order of steps, and I want you to give me the correct order of the steps, A through E. So pause the video here, come up with the correct order, and we'll go through it when we come back. Okay, the first thing that happens is DNA is transcribed into mRNA. Then mRNA attaches to a ribosome to begin translation. Then tRNA brings an amino acid to the codon from mRNA. Then the ribosome is going to move along mRNA to add amino acids to the peptide chain. And then finally, the completed polypeptide chain is released. This is transcription plus translation. Okay, that does it for today. We want to talk about mutations next and viruses last. So we just have a couple of small lectures left in this chapter, and then we'll be wrapping up chapter 17 shortly. So hopefully this made sense with codons. As always, ask any questions that you have in the comments below. Until then, I will see you soon. Bye-bye.